Dear viewers, in our previous video, we studied about prokaryotic cell. The link of that video I have shared in the description box of this video. So today we will study what is eukaryotic cell. This is part 2 of our chapter 8, which is cell, the unit of life. In this video we will study what is plant and animal cell, cell membrane. In cell membrane we will study fluid mosaic model, and finally we will study what is cell wall. So example of eukaryotic cells are protists, plants, animal and fungi. We know that plants and animal both contain eukaryotic cell, but they are very different in terms of their shape, size and the component which is called organelle present in them. For example, you can see here vacuoles, chloroplast and cell walls are present in plant cell, but these are absent in animal cell. Similarly, you can see centrosome, centriole, lysosome, peroxisome are present in animal cell, but these are absent in plant cell. And there are many components which are present in both cell. So here you can see cilium and microvillus is present, which is required for locomotory motion of animal cell. So it is very difficult to draw this structure on paper. So this is the easy way to draw plant cell and animal cell in examination. So in plant there is cell wall present, large vacuoles present, chloroplast present and flagella only in gametes present, but these components are absent in animal cell. So animal cell, they don't have cell wall, they don't have small vacuoles, no chloroplast, but they have flagella and trioles. And mitochondria, Golgi apparatus, endoplasmic reticulum, nucleus, cytoplasm and ribosome, these are present in both the cell. So this is the easy way to remember the component present in both the cell or absent in both the cell. Now here I have given table which differentiate between prokaryotic cell and eukaryotic cell. So first point is size. We know prokaryotic cell, they are small in size and it is around 1 to 10 micrometer. In eukaryotic cell these are large and it is around 10 to 100 micrometer. Regarding cell membrane, we know that prokaryotic cell membrane is covered with cell wall. If we talk about eukaryotic cell, animal cells don't contain the cell wall. And if we talk about organelles, most of the organelles are absent in prokaryotic cell, but in eukaryotic cell these are present, like mitochondria, nucleus and lysosomes are present in eukaryotic cell. And if we talk about nucleus don't have nuclear membrane in prokaryotic cell, but in eukaryotic cell nucleus are covered with nuclear envelope or nuclear membrane. Energy metabolism, we know mitochondria is the powerhouse of cell, but in prokaryotic cell mitochondria is absent. So enzyme required for energy metabolism, they are bound to membrane. But in eukaryotic cell, the enzyme required for energy metabolism are located in mitochondria. Cell division in prokaryotic cell occur through fission, but in a eukaryotic cell, it went through mitosis. If we talk about cytoplasm, organelles and cytoskeleton are absent in prokaryotic cell, but in eukaryotic cell, organelle and cytoskeletons are made up of tubules and filaments. So these are present in eukaryotic cell. Now we will study about cell membrane. So we know that electron microscope invented in 1950s. So after that a big revolution comes in the field of biology. It becomes very easy to see and study about the cellular component. But before that, scientists perform various chemical and biochemical reaction to study the component of cell. Scientists at that time, they majorly study about human red blood cell. And on the basis of that, they come with cell membrane composition, and it is made up of lipid, protein and carbohydrate. So if we talk about lipid, phospholipid is the main component of cell membrane. So it is arranged in the bilayer form. In phospholipid, there are two components. One is polar head, which is made up of phosphate chain. It is present outside of the cell membrane. And there is a hydrophobic tail, which is made up of hydrocarbon chain. It is present inside of the cell membrane. This tail portion is made up of saturated hydrocarbon, and its main role is to protect the cell from aqueous environment present outside of the cell membrane. Phospholipid membrane also contain cholesterol. So if we talk about protein there are two type of protein present. One is integral protein and other is peripheral protein. This is categorized based on their extraction procedure. Integral protein is very difficult to extract and peripheral protein is very easy to extract. Integral protein is embedded between phospholipid bilayer, while peripheral protein is present on the surface inside the cell membrane. If we talk about the composition, different cell is made up of different composition of lipid and protein. So if we talk about erythrocyte, lipid component present is 52% and protein component is present is 40%. And cell membrane model was first proposed by Singer and Nicholson and it is called by the name fluid mosaic model. So this is the cartoon of fluid mosaic model. So this is the phospholipid bilayer chain. This is one chain, this is another chain, and this circular portion, this is the phosphate group which is polar in nature, and this tail type chain, this is made up of saturated hydrocarbon, and somewhere cholesterol is embedded between this phospholipid bilayer. Here you can see a big protein is embedded between both the layer. This is called integral membrane protein. So it is difficult to extract this protein from this lipid bilayer. While peripheral protein is present on the surface, it is easy to isolate this protein. So easy way to draw the fluid mosaic model is like that. Here you can see this is the phosphate head which is polar in nature. This is a tail chain which is non-polar in nature and it is made up of fatty acid. Is there is saturated hydrocarbons are present, and you can see cholesterol is also embedded between this phospholipid bilayer. Here you can see there is a carrier protein present. It has its own function. It helps in transport of molecules which do not diffuse easily from this phospholipid bilayer, and there is some channel protein. Some molecules and ion transport from this channel protein very easily. So this is part of integral protein. And here you can see peripheral protein is present. So according to fluid mosaic model, nature of lipid is quasi-fluid. So movement of this protein is very easy in this cell membrane. The fluid nature of these cell membranes help in cell growth, formation of intracellular junction, secretion, endocytosis, cell division, etc. So the main function of this plasma membrane is in transport of molecules. So first one is passive transport. We know that cell membrane is very selective for some molecules or ion. So it allows them to diffuse through the cell membrane easily. So these molecules do not require any form of energy for their diffusion process. Know that osmosis is the diffusion along the concentration gradient. So some molecule from their higher concentration move to lower concentration. So there are some neutral solute molecule which use this osmosis process. Similarly, water move across the cell membrane from higher to lower concentration by this osmosis process. And finally, active transport. We know that in our cell membrane, there is non-polar lipid bilayer is present, which prevent diffusion of polar molecule through this cell membrane. So these type of molecules required carrier protein for their transportation. And similarly, some ion or molecule transport against the concentration gradient. So if you're moving some molecules or ion against the concentration gradient, you definitely require some kind of energy. So in cell, this energy they take it from ATP. ATP is adenosine triphosphate. So this process is called active transport, and there is like for transportation of sodium and potassium, there is sodium and potassium pumps are present in the cell. Finally, we will study cell wall. We know cell wall is the rigid structure present in the plant cell, and its main function is to protect the cell from mechanical damage or infection, and also it gives shape to the cell. It also helps in cell-to-cell interaction and provide barrier to undesirable macromolecule. Algae cells are made up of cellulose, galactans, mannans, and mineral like calcium carbonate, while plant cell is made up of cellulose, hemicellulose, pectin, and protein. So plant cell is made up of mainly three components. If you see, this is the plant cell. So this is the plasma membrane. Outside of plasma membrane, there is secondary cell wall. Outside of this secondary cell wall, there is a primary cell wall, and outside of primary cell wall, there is middle lamella present. Middle lamella helps to connect between two different cells. So this is the zoom out structure of cell wall. You can see this is the plasma membrane, and outside of this plasma membrane there is a primary cell wall. This primary cell wall is made up of cellulose and hemicellulose. You can see pectins are also present. So when they grow or mature, they become dye. So they create primary cell wall. Beneath this primary cell wall, secondary cell walls are present. And finally, this is middle lamella, which is made up of calcium pectate, and it worked as a glue between two cell wall. And there is plasmodesmata present. Plasmodesmata, you can see in this figure, it is a channel which allows cytoplasm to pass between two neighboring cells. So here guys, I finished in eukaryotic cell the component cell membrane and cell wall. In next video we will study few more components of eukaryotic cells. Thank you.